during meiosis, okay, we've got 46 chromosomes in a human cell. Recombination has occurred, and so if we look at these 46 chromosomes, I've just drawn 2 here. This is one homologous pair. And we see that recombination has occurred. That means on the blue chromosome, there's a little bit of pink genetic material, and on the pink chromosome, there's a little bit of blue genetic material, right? Meiosis 1 goes through prophase, metaphase, anaphase, telophase, divides, and it results in us going from one cell with 46 chromosomes to 2 cells with only 23 chromosomes in each. So we've taken the genetic information and we've cut it in half. Because if we don't cut it in half and you have an egg that has 46 chromosomes in it and it's fertilized by a sperm that has 46 chromosomes in it, how many chromosomes are you going to have? 92. Okay, I think it's like a muskrat or something like that that has 92 chromosomes. It's not you. So in order to ensure that your egg and your sperm cell, when they combine, they give rise to the proper number of chromosomes, you cut them in half. You cut the number in half. That's what meiosis 1 does. And then each of these cells is going to undergo another cellular division. It's going to undergo meiosis 2, so that it gives rise, when we have 23 chromosomes, we still have to go through one actual division to end up with 23 chromatids. So another meiosis 2 is going to occur with prophase 2. Prophase 2, metaphase 2, anaphase 2, telophase 2, and another cytokinesis. Okay? So that gives rise to 4 daughter cells. And in those 4 daughter cells, you are only going to have one of these 2 chromatids. So this is the one that had the genetic material that got swapped here. And then this one, it got swapped as well. And there we go. And that's the end result of meiosis 2. The end result of meiosis 2 is 23 chromatids, where you started with 46 chromosomes. Okay? So that's 4 times more genetic material that you had at the beginning. You had 4 sister chromatids for every pair, right? And now you're down to only 1 sister chromatid of each chromosome. So this first cell here, okay, has how many sister chromatids in it? How many sister chromatids would you have if you had 46 chromosomes? You would have 92 chromatids here. So we go from 92 chromatids in the first cell to 46 chromatids down to 23 chromatids. Okay? Or we go from 46 chromosomes to 23 chromosomes to 23 chromatids. Okay? And we have to do that in meiosis. Otherwise, the eggs and the sperm have too much DNA in them. All right? Now, if we have a non-disjunction disorder, which is what you actually have been working on, we looked at examples of Down syndrome. We looked at examples of Turner's, Klinefelter's, Patau syndrome, and Edwards syndrome. When you have non-disjunction, and you start off with your parent cell here with its full number of chromosomes, I'm just going to actually just draw 4 chromosomes in it, 2 homologous pairs. Okay, so I've got a big one here and I got a little one. Okay, and I got a big one here and a little one. So there's my full complement of chromosomes. Meiosis 1 occurs, and what should happen here? I should get, okay, a big blue and a little blue going in one side, and a big pink and a little pink going in the other side, right? Because we cut the number in half. But sometimes errors can occur. Okay, errors can occur in either meiosis 1 or meiosis 2. And if a non-disjunction disorder occurs, you have an uneven division of the chromosomes. So you can actually end up with a big one over here and a little one here, and maybe these guys didn't split up, but maybe these guys did. So we have an uneven dividing of DNA material. So if there's an error in meiosis 1, that means for sure there's going to be an error in meiosis 2, because we don't have the right number of chromosomes. These guys are going to keep dividing, right? We've got this guy who's going to split up, and this guy who's going to split up. Here, 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 and here. And then we're going to have 1x and 1x here. We're going to have 1x, 1x, another x, like this. Is that even? So if one of these are eggs, and one of these eggs gets fertilized with even a normal numbered sperm, the offspring will have either too much DNA or not enough DNA, and it will cause a genetic malformation. Depending on what the malformation is, depending on what the chromosome is, the outcome's going to be different. This is slightly different, though, if we actually have our parent cell up here with 1, 2, 3, 4, and then we have an even dividing in meiosis 1, right? So we have 1, 2, and then 3, 4. That's perfect so far. That's half. That's what we want. But meiosis 2 can actually give us a problem. Okay? So that can mean that we get, in one of these 4 cells, or 2 of these 4 cells, I should say, we get a problem. We could end up with this guy here and this, and only that there. And these guys could be totally normal. Okay? So these guys are normal, and these guys are abnormal. Well, if you're a female, and only 1 in 4 of these is going to actually give rise to being an egg, well, if you pick this one or this one, your egg's going to have a problem. If you end up having this one or this one, then it'll be fine. It'll be totally normal. And with women, more so than with men, as we get older, our meiosis messes up more frequently. Okay, so the incidence of non-disjunction divisions increases in females with it as they get older. So near the end of their reproductive lifespan. All right? And that's because women are born with all the follicular cells they're ever going to have. Your ovaries have all the follicles that they're ever going to have, unlike males who continuously make new sperm cells. Okay, we have all of these cells we're ever going to have. Roughly about anywhere from 400 to about 550 of them, depending on how long you menstruate for. Okay, because you're assuming you lose one every 28 days. All right? Whereas males, they keep doing this on a daily basis. Millions upon millions, even billions of times a day.